Good evening, wherever you are. This is Sophia Reyes from ConferenceHype. Our segment will focus on rethinking Alzheimer's. Novel mRNAs illuminate a disease beyond the brain. From the 2026 July edition of Molecular Psychiatry, Alzheimer's disease poses major health, social, and economic challenges to the modern world. Despite the advances in understanding AD, our knowledge about its pathogenesis remains incomplete. Recent data suggest that circulating microRNAs undergo complex changes in AD. Since these changes are yet to be comprehensively characterized, we investigated mRNAs in the context of AD using two meta-analytical approaches. We reproducibly identified 2895 mRNAs in a cohort of 4186 individuals from 22 studies. Here we show that 194 mRNAs exhibited widespread changes in AD, including some novel mRNAs not yet linked to AD. These novel AD mRNAs broaden the landscape of research on the role of mRNAs in AD. Targets of these mRNAs further uncovered many biological pathways that, to date, remain poorly understood in AD, with several AD mRNAs never described in the brain. From the 2026 July edition of Molecular Psychiatry, Rett syndrome is a neurodevelopmental disorder caused by mutations in methyl-CpG binding protein 2. MECP2 is a non-cell type-specific DNA binding protein, and its mutation influences not only neural cells but also non-neural cells in the brain, including vasculature-associated endothelial cells. Vascular integrity is crucial for maintaining brain homeostasis, and its alteration may be linked to the pathology of neurodegenerative diseases, but a non-neurogenic effect, such as the relationship between vascular alteration and RTT pathogenesis, has not been shown. Here, we developed a microvascular network model using RTT patient-derived induced pluripotent stem cells that carry the MECP2 or MECP2 mutation to investigate early developmental vascular impact. To expedite endothelial cell differentiation, doxycycline-inducible ETV2 expression vectors were inserted into the AAVS1 locus of RTT patient-derived iPS cells and their isogenic controls by CRISPR/Cas9. From the 2026 July edition of Molecular Psychiatry, 22 Q11-2 deletion and duplication carriers have an increased risk of neurodevelopmental disorders and exhibit altered brain structure, including white matter microstructure. However, the underlying cellular architecture and age-related changes contributing to these white matter alterations remain poorly understood. Neurite orientation dispersion and density imaging was used on mixed cross-sectional and longitudinal data to examine group differences and age-related trajectories in measures of axonal density, axonal orientation, and free water diffusion in 5022Q-DEL and 2422Q-DUPE carriers, and 890 controls. From the 2026 July 24 edition of Molecular Psychiatry, multiple psychiatric disorders are associated with later dementia, but it remains unclear whether these associations reflect a shared liability toward all psychiatric conditions or diagnosis-specific effects. In this Swedish register-based cohort study, we investigated these questions while adjusting for familial confounding shared by siblings. Exposures were 6 psychiatric diagnoses recorded by age 35 and a latent bifactor model fit to these diagnoses that identified 1 general and 3 specific psychopathology factors. Outcomes were all-cause dementia and Alzheimer's disease recorded after age 50. For observed psychiatric diagnoses, we estimated between individual and within sibling hazard ratios using Cox regression. For latent factors, we estimated between individual and within sibling odds ratios using exploratory structural equation modeling.
This is Conference Hype. Stay with us, coverage continues.
From the 2026 July edition of Molecular Psychiatry. While aberrant responsiveness to rewards represents a core transdiagnostic feature across attention deficit hyperactivity disorder and autism spectrum disorder, it remains unclear whether those deficits are underpinned by shared or distinct neurobiological signatures. Here we leveraged pre-registered task-based functional neuroimaging meta-analyses to identify shared and specific neurofunctional alterations during hedonic processing of rewards in ADHD and ASD, and further validated the distinctions by integrating behavioral decoding, brain network, and neurotransmitter linkage analyses. Our coordinate-based meta-analysis of 29 studies revealed that both disorders shared abnormal subcortical activation patterns in the putamen and amygdala, whereas the disorder-specific neurofunctional alterations were primarily localized to the prefrontal cortex. Such segregate neurofunctional patterns were further associated with differential behavioral manifestations, brain network connectivity changes, and anatomical expression of specific neurotransmitter systems. From the 2026 July edition of Molecular Psychiatry, magnetic seizure therapy and electroconvulsive therapy are effective treatments for schizophrenia and major depressive disorder, with MST offering advantages in preserving cognitive function. Although cognitive profiles are known to influence treatment outcomes, their role in moderating response to seizure therapies remains unclear. We conducted an open-label, non-randomized controlled trial to compare the efficacy of MST and ECT across different cognitive profiles. From October 2020 to June 2024, we recruited 263 patients with SCZ and 135 patients with MDD, identifying cognitive profiles through latent profile analysis. Among them, 54 patients with SCZ and 119 patients with MDD received MST or ECT, completing 3 or 4 sessions per week. MST was administered at 100% device output and 100 Hz over the vertex. From the 2026 July 24 edition of Molecular Psychiatry, rapid-acting antidepressants like ketamine and serotonergic psychedelics show promise for treatment-resistant depression, but the molecular mechanisms that contribute to their therapeutic effects remain unclear. Induced pluripotent stem cells offer a platform to model human cortical neurons and investigate drug effects in a human-relevant system. Here, iPSCs from individuals with TRD and healthy volunteers were differentiated into mature cortical-like neurons and treated for 6 and 24H with agents being investigated as rapid-acting antidepressants, including hydroxynorketamine, psilocybin, lysergic acid diethylamide, and 2,5-dimethoxy-4-iodophentamine. Bulk and single-cell RNA sequencing assessed global and cell-type-specific transcriptomic responses. Synaptic proteins were evaluated via Western blotting and immunocytochemistry. To validate translational relevance, transcriptomic results were compared to CSF proteomics from ketamine-treated HVs. From the 2026 July 25 edition of Molecular Psychiatry, autism spectrum disorder is a multifactorial neurodevelopmental disorder with complex molecular etiology. Since genetic causes account for less than 50% of ASD cases, novel approaches are required to overcome the challenges of characterizing genes and molecular pathways linking risk alleles to phenotype. Here we use RNA sequencing, three-dimensional protein-centric chromatin conformation, and whole genome DNA methylation sequencing, approaches to investigate hippocampal tissue from an ASD mouse model to determine if multiomic data integration improves the resolution of key molecular pathways contributing to the complex ASD phenotype. Each omic dataset individually identified disruptions in numerous genes and pathways, providing broad ASD-related insights, such as 699 downregulated genes with an enrichment of neuronal ontological terms, 2 unique chromatin interactions in CNTNAP2KO mice, and 3 that most differentially methylated genes have a neuronal function.
This is Conference Hype. Stay with us, coverage continues.
ConferenceHype is interactive AI commentary only. It is not reporting, journalism, medical education, clinical guidance, scientific validation, legal advice, or financial advice. ConferenceHype is independent and is not affiliated with conference organizers, presenters, sponsors, or exhibitors. From the 2026 July edition of Molecular Psychiatry, persistent mitochondrial inflexibility and mitochondrial damage may contribute to post-acute sequelae of COVID-19. However, data linking mitochondrial biomarkers, such as circulating cell-free mitochondrial DNA, to long COVID symptoms remain limited. We analyzed CCFMT DNA relative to glycerinaldehyde 3-phosphate dehydrogenase and total cell-free DNA in a nested case control study of 228 adults. Possible associations between these markers and general cognition, verbal memory, psychological distress, and inflammation were also examined. CCF-DNA, relative CCFMT DNA, C-reactive protein, and systemic immune inflammation index were assessed. Principal component analysis was applied to psychological tests to derive three components: general cognition, verbal memory, and psychological distress, which were used in further analyses. PASC patients exhibited significantly lower cognitive function and higher psychological distress than recovered controls. From the 2026 July edition of Molecular Psychiatry, major depressive disorder is a debilitating psychiatric disorder that is a leading cause of disability worldwide. Although treatment with antidepressants, such as selective serotonin reuptake inhibitors, has demonstrated clinical efficacy, the trial and error approach in choosing the most effective antidepressant treatment for each patient allows for only a subset of patients to achieve response to the first line of treatment. Circular RNAs are highly stable and brain-enriched non-coding RNAs that are mainly derived from the backsplicing and covalent joining of exons and introns of protein-coding genes. They are known to be important for brain development and function, cross the blood-brain barrier, and be highly sensitive to changes in both synaptic activity and neuronal receptor signaling. From the 2026 July edition of Molecular Psychiatry, Alzheimer's disease is a progressive neurodegenerative disorder characterized by a spectrum of cognitive impairments, ranging from mild memory loss to severe cognitive decline, and ultimately, death. The global incidence of AD is projected to increase significantly, with late-onset AD being predominantly sporadic in nature. Over the past 3 decades, the apolipoprotein E gene has been recognized as the most important single genetic determinant of sporadic AD risk. The APOE4 allele is a major risk factor for AD and is known to exacerbate the pathological process for AD. Identifying protective variants that may reduce the risk or delay the onset of AD is of great significance for the development of effective treatments. This review comprehensively examines the protective effects of APOE and its related protective mutations. From the 2026 July edition of Molecular Psychiatry, esketamine has emerged as a fast-acting antidepressant option for individuals with treatment-resistant depression. Yet, little is known about how sex assigned at birth shapes symptom-specific responses to these interventions, a critical gap in the move toward precision psychiatry. To address this gap, we conducted a pooled analysis of 5 randomized, double-blind, placebo-controlled trials in which adults with TRD received intranasal esketamine or placebo twice weekly for 4 weeks, alongside a newly initiated oral antidepressant. We evaluated the effects of sex assigned at birth on overall depression severity, measured via total Montgomery-Åsberg Depression Rating Scale scores, across 4 symptom factors: sadness, negative thoughts, detachment, and neurovegetative symptoms, and rates of clinical response and remission. Esketamine treatment improved total MADRS scores in both sexes.
This is Conference Hype. Stay with us, coverage continues.
That's it for now for Conference Hype's coverage of the July 2026 issue of Molecular Psychiatry. If anything was missed, tell us which article deserves follow-up. Tag us on X. Add your perspective in the comments, share this review with your clinical team, like the video, and subscribe with notifications turned on for the next journal broadcast.